Okay. Good morning, everyone. Myself, Dr. Kosta Fatima from Osmania Medical College. My title of my study is Detection of Extended Spectrum Beta-lactamase and Metallo-beta-lactamase Production in Pseudomonas aeruginosa at a tertiary care center. Pseudomonas aeruginosa is a gram-negative opportunistic pathogen responsible for a wide range of nosocomial infections, including surgical site infection, septicemia, urinary tract infection, and the lower respiratory tract infection. The nosocomial infections caused by multi-drug resistant organisms have become a major clinical and public health concern. The resistance to broad-spectrum beta-lactamase is an increasing problem worldwide, which is mediated by extended spectrum beta-lactamases, AMP3 beta-lactamases, and metallo-beta-lactamase enzymes. Carbapenems are the last line of defense against many drug-resistant bacterial infection. Unfortunately, infection due to carbapenem-resistant pathogens are on the rise. The production of beta-lactamases such as ESBL and MBL are the most common resistant mechanism documented in Pseudomonas aeruginosa. The aim of study is detection of ESBL and the MBL in Pseudomonas aeruginosa isolates at a tertiary care center. The objectives are isolation, identification, and susceptibility testing of Pseudomonas aeruginosa from clinical samples, detection of extended spectrum beta-lactamase production in Pseudomonas aeruginosa, detection of metallo-beta-lactamase production in Pseudomonas aeruginosa. The inclusion criteria are samples from the ICU, burns ward, medical ward, surgical ward, both the sexes, age group 16 to 60 years, and participant giving consent. The participants which are not giving consent and immunodeficient persons are included in the exclusion criteria. It's a cross-sectional study to isolate, identify, and susceptibility testing of Pseudomonas aeruginosa from clinical samples received at Microbiology Laboratory, Osmania General Hospital, to detect the production of ESBL and MBL among the isolates by phenotypic methods. The study was conducted over a period of 6 months from January 2021 to June 2021. A total of 327 specimens include exudate, urine, sputum received at Microbiology Laboratory at Osmania General Hospital. Culture of all the samples were done on the blood agar and McConkey agar and incubated at 37 degrees for 48 hours. The isolates that showed non-fermenting colonies of McConkey agar were identified using gram staining morphology, catalase test, oxidase test, and motility by hanging drop method. The microbiological identification was done using biochemical tests like indoor production, citrate utilization, urea hydrolysis, and triple sugar iron. Antimicrobial susceptibility by Kirby-Bordis diffusion on the Muller-Hinton agar following Central Laboratory Standards Institute Guidelines 2020. The isolates which were resistant to beta-lactam antibiotics were considered as beta-lactamase producers and further tested with combined test with ceftazidime and ceftazidime with clavulanic acid. And the isolates which were resistant to imipenem considered as carbapenemase producer and further tested with combined test imipenem and imipenem with EDTA and modified carbapenem inactivation method and EDTA modified carbapenem inactivation method. Detection of ESBL, the requirements are ceftazidime with 30 micrograms, ceftazidime with clavulanic acid 30 by 10 microgram, Muller-Hinton agar plate, and the test isolate. The lawn culture of 0.5 methylene standardized test isolate was done on Muller-Hinton agar plate and 30 micrograms ceftazidime with and ceftazidime with clavulanic acid placed 20 mm apart from H2H and incubated at 37 degree for overnight. The more than 5 mm increase in the zone diameter of the ceftazidime with clavulanic acid when compared to the ceftazidime alone is considered as ESBL producer. Detection of MBL by combined disc diffusion test. The requirements are imipenem 10 microgram, imipenem with EDTA 10 by 750 microgram, Muller-Hinton agar plates, and the test isolate. A lawn culture of 0.5 methylene standardized test isolate was done on Muller-Hinton agar plate and 10 microgram imipenem disc and imipenem with EDTA disc placed 20 mm apart from H2H and incubated at 37 degree for overnight. More than 5 mm increase in the zone diameter of imipenem with EDTA disc when compared to the imipenem disc alone is considered as MBL producer. The modified carbapenem inactivation method and EDTA modified carbapenem inactivation method ECIM, the requirements for which are 30 K soy broth, Muller-Hinton agar plate, Escherichia coli ETCC 2.922, meropenem disc 10 microgram, 0.5 m EDTA solution, and test isolate. The Pseudomonas aeruginosa was grown on the Muller-Hinton agar plates, 10 microliter of which from the overnight grown plate was emulsified in 2 test tubes of 2 ml 50 K soy broth, which were labeled as TSB1 and TSB2, vortexed for 15 seconds, and then for MCIM, 10 microgram meropenem disc added into the suspension in TSB1 using a sterile forceps, ensured the disc is immersed in the suspension completely. For ECIM, 20 microliter of 0.5 m EDTA added to the TSB2 to obtain a final concentration of 5 mm EDTA. Then a 10 microgram meropenem disc into the TSB2 using a sterile forceps. Both the tubes incubated at 37 degrees for 4 hours. Lawn culture of 0.5 Macfarlane adjusted culture of equally ATCC 2.922 done on Muller-Hinton agar. Meropenem disc from the TSB1 and TSB2 tubes were placed on the same MHA plate, inoculated with a meropenem susceptible equally ATCC 2.922 indicator strain. The plates incubated at 37 degrees for 18 to 24 hours. Following the incubation, the zones of inhibitions were measured. The interpretation of the MCIM is it is considered as carbapenem is positive if the zone of inhibition of the E. coli is 6 to 15 mm and negative if the zone of inhibition is more than 19 mm, and it is carbapenem is intermediate if the zone of inhibition of the E. coli is 16 to 18 mm. The interpretation of the ECIM is it is considered as metallo-beta-lactamase producer if there is a more than 5 mm increase in the zone diameter for ECIM disc versus the zone diameter for the MCIM. If the test isolate produces a metallo-beta-lactamase, the activity of the carbapenemase will be inhibited in the presence of EDTA such that the meropenem in the disc will not be hydrolyzed as efficiently as in the tube without EDTA. The result is inhibition of the meropenem susceptible E. coli and an increase in the zone diameter for the ECIM when compared to the zone diameter of MCIM. It is considered as metallo-beta-lactamase negative if there is less than 4 mm increase in the zone diameter for the ECIM versus zone diameter for MCIM. If the test isolate produces a serine carbapenemase, the activity of the carbapenemase will not be affected by the presence of EDTA and there will be no or marginal increase in the zone diameter between the two discs. The results of my study are a total of 85 Pseudomonas aeruginosa were isolated from 327 different clinical samples. This is the sample-wise distribution of Pseudomonas aeruginosa from the different samples. The maximum number of Pseudomonas aeruginosa were isolated from the pus. Among the 85 isolates of Pseudomonas aeruginosa, 32 were ESBL producer by combined disc diffusion method, that is accounting for 37.6%, and 27 were MBL producer by MCIM and ECIM method, that is 31.7%. Among the total 27 MBL producing isolate detected by MCIM and ECIM method, only 20 were detected as MBL producer by combined disc diffusion method. ESBL and MBL producing Pseudomonas aeruginosa continue to be a major challenge in healthcare institution and accounts for 10 to 50% depending on the local epidemiology. Resistance is mediated by a variety of mechanisms including drug efflux, alteration in the membrane permeability, and the production of beta-lactamases such as serine and metallo-beta-lactamases. Hence, knowledge about their prevalence is very essential to initiate appropriate antimicrobial therapy. The MCIM and ECM test is phenotypic method of choice to distinguish MBL carbapenemase and serine carbapenemase in Pseudomonas aeruginosa. This is the ESBL and MBL detection in prevalence in the different studies. In the present study, the prevalence of the ESBL is 37.6% and the MBL 31.7%, which is similar to the study done by Gomati et al. In 2016. The ESBL prevalence is 18.18% and Aruna et al. In 2018, the MBL prevalence is 53.8%. Timulay et al. In 2019, the ESBL prevalence is 42.6% and the MBL prevalence is 40%. And that S. Tiwari et al. In 2020, the ESBL prevalence is 26.09%. In the present study, maximum number of Pseudomonas aeruginosa isolated from the pus, that is 64%, similar to the study done by Tiwari et al. 2020, who reported the maximum number of Pseudomonas aeruginosa from the pus and swabs, 35.7%. Sachdeva et al. In 2017, in their study, to know the prevalence of MBL producer by different phenotypic methods, they found that the combined disc test has the highest sensitivity and specificity, that is 97.9% and 96.1% respectively for the detection of MBL production as compared to other tests. But in the present recent study by C.M. Gill et al. In 2020, who reported the modified carbapenem inactivation method successfully categorized the majority of the Pseudomonas aeruginosa isolates, resulting in a sensitivity of 93% and specificity of 85%. The EDTA carbapenem inactivation method testing procedure was able to differentiate VIM and NDM producers from the serine carbapenemase producer in diverse collection of clinical Pseudomonas aeruginosa isolates. Simner and colleague in 2017 evaluated several phenotypic methods for identifying carbapenemase production and reported an MCIM test has sensitivity and specificity of 100% and 98% respectively. To conclude, the prompt and accurate detection of ESBL and MBL production in Pseudomonas aeruginosa is crucial for optimal treatment of critically ill and hospitalized patients and also to control the spread of resistance. The MCIM and ECM test is described as more advantageous over the other phenotypic tests for MBL detection as it detects the MBL carbapenemase and serine carbapenemase in Pseudomonas aeruginosa and has sensitivity and specificity of 100% and 98% respectively. These are my references.